Hello, my name is Hannah Davis from Hayti High School, and my project is entitled An In-Depth Look into Identifying the Differentiation Pathway and Cell Type of the Dual Receptor Expressing Lymphocytes in Type 1 Diabetes. A little bit of background information about my subject. Type 1 diabetes is an autoimmune disease in which the body attacks its own islets of beta cells within the pancreas. The most common treatment for type 1 diabetes is insulin injection, and the average sufferer spends between 6,000 to 7,000 yearly on insulin alone. Diabetes is a disease of the immune system, which is the body's first defense against intruders known as pathogens. The immune system can be divided into the passive, active, and innate responses. Within these responses, you're going to have cells known as lymphocytes that officiate immune response. However, when the immune system does not work as planned, the immune cells target the body's own functioning cells as faulty cells and marks them for destruction. One new cell found in relation to type 1 diabetes is the DE cell. The dual receptor expressing lymphocyte is named because it displays both a T cell receptor and a B cell receptor, and according to the article in which the cell was originally published, these cells are shown to mimic insulin peptides to trigger diabetes-specific helper T cells. And although it could be supported that the DE cells are driving type 1 diabetes, this was not explicitly stated in the article that these cells were originally published, and it could not be adequately concluded by other scientists researching this topic. My project, in order to interpret the data, I'll be using bioinformatics, and within bioinformatics I'll be using RNA-seq analysis, and within that I'll be using differential gene expression analysis. So the purpose of my research is to provide as much information as I can about the DE cells to hopefully provide insight to see if these cells actually cause type 1 diabetes and what their role is in causing the disease. Furthermore, identifying the cell type and differentiation pathway of these cells could provide insight into other diseases as well as prevention and reversion methods for type 1 diabetes itself, which currently do not exist. So the materials that I used, first I used the NCBI SRN table from this bio project right here. And I chose this data set because it's the original data set that the DE cell was originally published, and the name of the article is a public BCR present in a unique dual receptor expressing lymphocyte from type 1 diabetes patients encodes a potent T cell autoantigen if you're looking to find that on NCBI. And I also used a computer with the capabilities of running my bioinformatics platform. I used the Tauber Bioinformatics T-BioInfo platform for RNA-seq analysis provided to me from Science Coach. And I also used Microsoft Excel to interpret my data. Okay, for my procedure, first I downloaded the SRA run table from the bioproject in question. Then I input the files into my platform, selected 2 cell types with 3 samples each to compare at a time. Then I ran a pipeline. Specifically, I chose the top hat cuff merge, cuff diff pipeline for differential gene expression analysis because it output a normalized expression count, which would be better indicative of the expression of the set as a whole since I only used 3 samples. And then I downloaded the gene expression output files and drew conclusions based on these outputs. So here's an example of some of the graphs that I made. This is the DE cell's top 50 expressed genes. Specifically within this, I'd like to point out the expression of this natural killer granzyme protein, as well as this immunoglobulin heavy protein being in the top 50, as well as a couple of these mitochondrial RNA genes being expressed, which would indicate a high proliferation rate of this cell. Here's the natural killer gene signature being expressed at very high rates by the DE cell, but not so much by the T and B cell. And to note that the NK cell and T cell are very similar in nature. However, the DE cell is showing that it is closer to a natural killer cell rather than a T cell by expressing more of these genes. Specifically, I'd like to point out perforin and some of these chemokines that are being excreted. This is the B cell gene signature being expressed by both the DE cell and the B cell alike. Here you can see that the DE cell is expressing cytokines as well as the TCR. So in conclusion, the DE cell is going to possess lineage markers indicative of a CD8 NKT cell, which suggests that the DE cell has B cell features because of its germline encoded BCR. And I was able to conclude this because in the original article it stated that the BCR on the DE cell is of germline configuration, and this supports the idea that the DE cell is a dual purpose cell. So this leads to the differentiation pathway. So you can see that the DE cell is going to follow a relatively standard differentiation pathway of a natural killer cell. However, within TCR activation, it's going to be activated by self-antigen binding, and it's going to stop at the naive NKT cell and not develop beyond that. And after that, it's going to travel back to the bone marrow, where its BCR is going to become activated, again by self-antigen binding is what I hypothesized. And I concluded all of this based on the expression of different cell markers being expressed, as well as the different TCR and BCR genes being expressed. So this is the proposed cell function model. Here you can see the DE cell, and it's going to identify this beta cell islet through either BCR antigen identification or that insulin peptide mimicking, as mentioned in the previous article. However, either way, it's going to secrete cytokines in the form of Th1 and Th2 signaling, which will lock to this TCR right here, which will trigger cytotoxic activation in the form of granzymes and proferrin, which are being expressed at a very high rate by the DE cell. And that's going to attach to the beta cell islet and trigger apoptosis. So what does this mean for autoimmunity and type 1 diabetes? Well, a cell of this type and function has never been explicitly identified, and if the cell function model that I have proposed and the conclusions I've gathered are correct about this cell, then that means that this cell is a prime candidate for being the #1 driving factor of type 1 diabetes. Furthermore, a natural killer T cell and a B cell with B cell features indicates a much closer connection between the innate and adaptive immune system than previously described. And it is very plausible that this mutant lymphocyte could be in and actively contribute to other autoimmune diseases and even cancers, and the future of treatment relies on more research being presented regarding these cells. Thank you for taking time to listen to my presentation.